Well, thank you very much, William, for that very effective and good introduction. And thank you to you for coming today. It's great to see this lecture theatre full. As William said, just over 100 years ago, Dr. Dunstan gave a lecture at the Fruit Goers Conference in the Town Hall at Maidstone that led to the conception of East Malling Research. And this is the final, as he said, the final celebratory activity of our 100 years. And what I'm going to do today is very briefly summarise for you what Dr. Dunstan said and the consequences in relation to what was, I'm going to call it East Malling Research, EMR throughout, okay? It's had many names over that 100 years, but we're going to call it EMR today. So I'll summarise what that led to. I'll talk about the context and content of some of our present research before I spend, I hope, most of the lecture, if I don't run out of time, talking about the future and what the future brings. Now, if you look at what Dr. Dunstan said, and there's a very good summary actually in a book that we have, the original typescript of the meeting. If you look at what Dr. Dunstan said, these are his main points. And his main stress at that time was on what he called the individuality of the fruit tree. He pointed out that trees in an orchard were all different shapes and sizes. He pointed out that, as I've put it here, the growth of the wood opposed its fruition. There was clearly a lot of concern at that point about how you both got the tree to grow and the fruit to grow. And he pointed out the importance of the choice of the scion, the top part of the tree, and the rootstock. And he then went on to talk about these other points here, the non-setting of blossom and pruning, the importance of those. He talked about insects in particular and fungi, and a curious thing, obviously winter washes and so on, although regularly applied, often had adverse effects on the growth of the tree. And he talked about the need to have new varieties and extensive testing. If you move on a year or two and look at what, in fact, the next couple of directors did and what they said, the impression that you get at that point is very much of local issues, the focus on the local industry and issues that were particularly important here in southeast England. Not surprising, because of course there was a fruit research station at Bristol at that time, at what became Long Ashton, and there were other fruit research endeavours up in East Anglia too. So the focus was very much on the local. The mission is as stated in the central point there, and this mission, as stated by Captain Wellington very early on in the life of this institution, has continued to this day. We are concerned with real problems in the actual culture of trees and bushes. And interestingly, almost a modern phrase, they gave priority to big issues, complex problems, knotty problems, I suppose we'd say this day, big issues, and they chose to work on rootstocks. And we know, of course, that concentration of resources on big issue brought very effective results. And this slide here shows the effect of different scions on the same variety of apple, that the scion size is controlled by the rootstock. And that, of course, among other things, is what has made East Malling famous around the world, and this technology is still in use today. So that's the past, and that's where I'm going to leave the past, and I'm now going to move immediately to today. What's the context today, if I were trying to persuade you that we should be set up? Well, the context today can be summarised really by these two documents. The one on the left is a document published early in 2010 by the Chief Scientist's Office. It came out of a thing called the Foresight Report, and it's called The Future of Food and Farming. This was 3-4 years ago now, a major document. Why? Because for most of my career, it was not a polite topic of dinner time conversation to talk about food and problems with food. It was a real turn-off as a party conversation piece, talking about that. Now, in the space of a few years, it's become recognised actually that food security and the way we produce our food is back on the agenda as a big issue. And last year, in July, was actually launched here at East Malling a UK strategy for agricultural technologies backed by new money from government to try and revitalise elements of our industries and to make us internationally competitive again. The document points out, in particular, how we have in the UK some absolutely first-rate plant science, but it's not finding its way into application because we have largely done away with all of the public sector institutions that ensured that translation. And of course, we are one of those organisations that is no longer in the public sector. However, we're still doing a lot of the translation. So that's the political context. And now is, as I think, at the close of my career, is as good a time as any to be embarking on a career in this area. The other contexts, of course, have changed. Food production, I've mentioned, and food security, that's gradually, actually, currently, moving into much more of a focus on nutrition, and I'll come back to that later in my lecture. There's a great emphasis now on using resources efficiently. That's tied up with so-called sustainable intensification. And those interests also ally with the importance of reducing waste. Climate change and variability is on the agenda, and I'll refer to that again later. And the other thing, of course, which has changed enormously in 100 years, is that we are now part of a global industry. And the problems, many of the problems that we look at are not just local in nature, but global and have applications beyond these shores. So we're a global industry, but with premiums, and this is the challenge really, but there's much more of a pure premium for local, authentic produce. Produced in Kent is serious. And that's not just true of produced in Kent. Local authentication is now a big issue, and people are prepared to pay for it. And the other thing that's changed, of course, at a number of scales, is that we're increasingly serving an urban population who isn't particularly interested, actually, in the details of how their food is produced or gets to them. But I'll come back to that point again later, too. Now, scientifically, the context for what we do, I always think it's useful to have a sort of a conceptual framework for your work. It'd be interesting to ask the scientists who are working here what they think that is. But I think it's this. I think it's very simple, actually, and it actually comes from arable agriculture. If we were wheat producers, we'd have this very much at the forefront of our minds. And this is what we do. We know the production really comes from the interaction, the function of, and the interaction between the genotype that we choose, the environment, and the management that we care to give the crop. And that's what we do. It's a gem. That's what we do here at East Malling, and I'm just going to give you a brief idea of one of each of these types of research and what we're currently doing. So I'm going to start with genetics, the genotype, and of course here we produce new varieties. Now this is a very, very exciting time to be involved. If you're a plant scientist, it's an incredibly exciting time because we now have the ability to look at the genetic structure of many of the plants and indeed insects and so on that we're interested in. And this is a summary of some work that we were part of a few years ago as part of an international consortium to get the genetic structure, which is called the genome, of Fragaria vesca. That's a wild strawberry. This wild strawberry is easier to work on than the domesticated strawberry. So we've got now the genome, and what you're looking at here is two things. On your left-hand side here, you can see that there are 7 chromosomes, and these horizontal bars here indicate genes that have been identified around on these 7 chromosomes. And then on the right-hand side, as you look at it, is a reconstruction of a whole number of different plants in the Rosaceae family. So that's apples, other fruits of all sorts, and roses themselves, of course. And you can see here that it's shown on 8 different chromosomes. And what you can see is that a lot of these a lot of these genes here, over here, ally to other things in the Rosaceae. You'll see that if you look at it this way around, that some of the things, for example, which appear on chromosome 2 in many of the Rosaceae have been shuffled to appear on chromosome 7 over here. But a lot of genes, which we know about here, are also common in this particular strawberry. The shuffling has gone on. But if you look at it the other way round, you see that many of the things that we know now about this strawberry in detail can be applied to other Rosaceae. And so we have a model here, a model that we can use to infer about the performance of other members of the Rosaceae. And that is incredibly useful and important piece of research to us. So how are we actually using it? It's just one example to start with. So, particular problem, verticillium wilt in strawberry. Here is a strawberry that is tolerant to the verticillium wilt. Here's one which is on death's door. And the way we can go about using this and exploring this is to cross these two. So you cross something which is very tolerant with something which is very susceptible, and you get the children. You get the children plants, and we get 200 or so seedlings that will vary greatly in their susceptibility to this fungal disease. And that's called a mapping population. Of course, you can't do that with humans. You never have 200 of them to look at. But you can with things like strawberries and plants, and this makes it quite exciting. So we can then start to look at, okay, if we find something which is tolerant, let's have a look at the genes and show which ones show up. And in this particular case, in 2 years, in 2009 and 2007, you can see that we've got this big peak here corresponding to this gene here, which suggests that it's probably involved in that resistance. Complication is that many diseases, and verticillium wilt is one of them, are not just regulated or controlled by single genes. And if you do that experiment and you repeat it over 3 years, for example, you can see that we get what are called these identified loci showing up in different places from year to year, but some are consistent. This one is here in all 3 years, this one in 2, this one in 2, and this one in 2. But because it's multigenic, others show up depending upon the season. And when we go back to the field and look at our scores, so this is something down here which is completely tolerant or resistant, and this is something which is very susceptible. We find basically the more markers that we've got, the more of those peaks that we can identify, the more tolerance or resistance we've got, but there's quite a difference from year to year. And we're working at the moment on why that is. So these genomes and this power of genetics now is a very significant contributor to our research. To celebrate our and we can now use that, and it's now being used in our breeding program, and we are starting to use that technology to identify at a very early stage resistant lines so that we don't have to plant them out for year after year. To celebrate our 100 years, we thought that it was about time we took the power of that genetics to the rootstocks. because unbelievably, nobody had got full sequences for the common rootstocks. And we were very fortunate that the HDC panel, Tree Fruit Panel, agreed to fund some of this work. And this is at a very early stage, so we haven't got that genome that I showed you earlier, but we have got M9 sequenced, and we have been able to assemble it. These are currently being sequenced, but already we're starting to get some interesting results from this work. So this is what's called a phylogenetic tree. It shows all the different sorts of malus that there are. And what's interesting is that our M9 rootstock sits up here alongside Malus domestica, the common apple, and Malus orientalis, which shows that our rootstock here, our M9, has come from, as it were, or inherited or got within it genes that are common to Malus domestica and Malus orientalis from Central Asia. It will be interesting to see where our other rootstocks come from. Moving on to management, an example of some management, and I've just picked one example here, some work that's going on involving some very smart biology and some very smart chemistry, collaborative work between ourselves and the Natural Resources Institute at the University of Greenwich. A particular problem in apples, as many of you will know, and that I know from my back garden, is the apple leaf midge, which produces the effect that's shown here, these curled up leaves, and if you open up the leaves, here are the little weevils sitting inside the leaf. And there is no effective control for this apple leaf midge, and if you get it, and it can do a considerable amount of damage. So the work that's been going on here has been to try and find a way of monitoring and trapping this insect. And what Jerry and the team have done is to collect a whole lot of, in fact, reared under controlled conditions, over 10,000 virgin flies. And then these virgin flies have been put into this piece of apparatus here, and air passed across. And you can see the little virgin fly here with her ovipositor. She's just ready to lay eggs. And at that point, she needs a mate. And she releases a pheromone, a chemical, to attract a mate. So in this, what has been done here is to put the virgins in here, pass air across them, and capture the very, very small amounts of chemical that this virgin fly is releasing. You then do the tempting experiment, where you place the male here. You place like a heart monitor, but it's like a cardiogram, but on the antennae. Measures the electrical impulses generated by the antennae, and you waft across the antennae those chemicals coming from the virgin fly. And if you do that, as here, you see there's no response at all for a time, and then suddenly, bang, a response. And if you ally that with some very good chemistry, this is using a gas chromatogram, these are the chemicals that are being passed along. And you might think, oh well, it would be the chemical that's in the highest concentration that elicits the response, but it's not. It's actually this one here. And again, you can do some analysis then to focus on that particular peak that I showed you earlier, break down which chemicals it is that are giving that response, and that puts you in a position then to produce a lure to lure the male flies to a monitoring device shown here. So this is the chemical. This is what is actually attracting those males to the female, and it can be put into a little vial here. The flies are attracted. You can't call this a trap, because under European regulations that would require you have to go through a lot of regulatory hassle to track the things, so we monitor them. The fact that they stick to the paper and die is incidental. So here are the traps. There's other work involved in this as well. It's very crucial where you site the trap and so on, the height. You might think, okay, this is an apple midge. I showed you the tips of the leaves. You'd put the trap up by the tips of the leaves. Wrong. You put it down near the soil surface, because that's where the flies are waiting for their mates. So there's quite a lot of other work involved in this, but the guts of it is clever chemistry, clever biology. And that enables the management of this particular midge and others too now. And finally, the environmental part and using the resources efficiently. One of the resources, obviously, that we use in producing crops is water. And I'm going to show just an example from Mark Else and his team. We know that many of the areas here in Kent, over here in Warwickshire, Staffordshire, here in East Anglia, the areas where we are producing fruit are prone to water shortage. And 3/4 or so of UK soft fruit growers are growing in water-stressed areas. By and large, growers use too much water to produce their fruit. And they do that for good reasons. Basically, when I was a university lecturer, I used to describe it as the whiskey issue. If you have one glass of whiskey, it makes you feel really good. So you're tempted to have another, and another, and then you feel really lousy. And that is, to a large extent, what we're doing when we're using water in many of our crop production facilities. We're using too much. So, So growers like to see this soil looking nice and dark and moist, whereas in fact, we could grow our crops on soil which is much less moist and still have no effect on yields. And the reason for that is, and this is something which shows basically the wetness of the soil. This is wet soil, this is drier soil. What we find is that as the soil dries, it starts to affect different processes in the plant. So one of the first things it affects is the stomata. Then it affects a thing called basically the strength with which the water is held within the leaf. Then it starts to affect photosynthesis, then the leaf extension rate, and then the fruit extension rate as the soil gets dry. And in commercial practice, of course, you don't want any of these things to happen, so you keep the soil wet up here. But we could actually, as you can see, let things be a bit drier without affecting any of these processes. We want to have a bit of a buffer zone before we get into mild water stress, and we certainly don't want to be down here. But up here, there's quite a margin that we can exploit. And that's essentially what Mark and his team have been doing. Could we regularly grow sort of in this region rather than up here? And it would save water, but it would have other beneficial effects too. So we can get savings in water, 36%, important perhaps in a dry year, not necessarily economically important because we don't attach much economic value to water at the moment. But also you can save fertilizers. There are no nutrient deficiencies. Mark and the team have shown that we can get improvements in class 1 yields and in shelf life. And these things, this and this, are economically important as well as important to production. And this work is now being rolled out with a number of growers. So that's where we are at the moment. I put this slide in because it also shows another characteristic of research in this day and age, and that is we work with multiple partners, both from the public sector and across the private sector. And Mark and Jerry and Zhao Ming, who head up the team, spend a lot of time working with our partners, and these are just some of them. And you can see that the effect, basically, that this research has had. This is the amount of water to produce a strawberry using typical grower ways of doing things. And this is, as it were, the minimum amount of water that you can use to produce that strawberry. So it shows the potential there for significant savings of water. And this work is now being rolled out, as I've said, with soft fruit growers in this country, and interestingly, with rose growers in Kenya. So what about the future? What does the future look like? Well, of course, nobody can predict the future, so what I'm about to say is almost certainly going to be wrong in total. But I think there are things in it which will not be wrong and build on trends that we can already see. And I've picked out 4. There are others, of course, but I'm limited in time. We need, the buzzword at the moment is increasing production sustainably, so sustainable intensification of production and with low waste. Climate change and variability is an issue, and I'll say a bit about that. I'll talk about environmental control, and then I'll say something about nutrition. And the feature of, I think, all of these topics and the significance for East Malling research in the future is that these will require multiple disciplines. These are difficult problems, back to what Dunstan identified. These are the difficult problems of this day and age that will need multiple disciplines to tackle, in addition to these classical disciplinary approaches which I've talked about already. And this move towards multidisciplinary teams is already happening here at East Malling. So, again, in a reflection of Dunstan's lecture, Dunstan gave his lecture basically saying, look what Rothamsted's done for arable research, we could do the same for fruit. Again, if we were arable specialists and arable scientists, we'd be very familiar with 2 concepts that I've put on the screen here. And these are important concepts, actually, for horticultural crops as well. Basically, what we do when we produce crops, what we're looking to do is to intercept the sun's energy, the radiant energy from the sun, and convert it into chemical energy in a plant, into sugars, and to grow a plant. It's very simple, actually. We're capturing energy, and we're making it into plants. And the amount of dry matter that we get is equal to the amount of radiation that comes from the sun, the amount of that sunlight that we intercept with leaves, and then the efficiency with which we convert that radiant energy into dry matter. And this concept has been around now for about 40 years. And it's important because if we know these terms, we can calculate what the potential production should be. Now, we're not just interested in all the dry matter. Dunstan, of course, quite rightly, was interested in the growth of the tree and the growth of the fruit, but most of us are interested in just that harvestable bit. And so that's the dry matter that we've produced here. And then it's the amount of that dry matter, called the harvest index, that goes into the fruit. And that's where, if we were wheat producers, that's where we'd stop. For horticulture, we have to add an extra term, which is the fraction of that which is going to be marketable, the class 1 or the class 2 or whatever it is, because not all of the fruit is going to be marketable, and it's important that we know about this. Now, these concepts, I think, are extremely important, because if we're going to talk about, as we're being encouraged to, sustainable production, then we at least ought to have some idea, some quantitative idea, of what it is we're aiming for. There's no point in saying, well, we're going to increase production if actually we're already at the limit. And how will we know whether we're at the limit? We'll only know if we take on board concepts like this. And I just want to explain that. So we've known for a long time, this is actually a paper that was produced in the first year of my postdoc by my supervisor, John Monteith, who was a physicist. And what John Monteith showed in a very famous paper was that actually apples, and indeed all crops, are very similar. The amount of dry matter that they produce is related to the amount of radiation that they intercept, And basically, apples, barley, potatoes, and sugar beet all produce 1.4 grams of dry matter per megajoule of radiation intercepted. There are variations, slight variations, but basically they all do the same thing. Now that's important. That's very important, because actually it means that those concepts and the other research that's been done on other crops can also be usefully used in fruits. And indeed, I just pulled this out from the literature. This shows apple yields. It's not quite the same as the previous graph. It basically shows the amount of light that's being intercepted in the middle of the growing season. This is a whole set of experiments from the UK, the USA, and the Netherlands, and it shows that yields of apples are related to the amount of light that's intercepted. But of course, we're interested in a harvestable yield. And going back to Dunstan's comment that the tree and the fruit are in competition, they are, because if we basically partition too much of the dry matter to fruit, the size of the fruits starts to go down, and they're not marketable. Now, I can find very few examples, or I can find very few examples of what this relationship looks like for different varieties, but you might reasonably expect that it would differ between varieties. But finding out what this relationship is for modern cultivars seems to me to be really quite important. Now, what this all means is, if you put all of these things together, and the best example that I can find currently of this comes from a paper produced in New Zealand in 2002, where they grew Braeburn and Fuji, and they plotted the amount of dry matter that was produced against the amount of light intercepted. John Palmer had got results from the UK here, which is what's shown in the squares, and all the New Zealand results are shown in the black. It's immediately obvious that the New Zealand yields are higher than those in the UK. That's principally down to the fact that they get more sunlight there during the growing period. And what this, if you put into this that harvest index term that I showed you earlier, if you allocate roughly 70% of the dry matter to fruit, if you intercept 90% of the radiant energy coming in, that incidentally is exactly the value that wheat farmers in this country are aiming to do. They're basically, if you're a wheat farmer, you aim to get your crop to a leaf area index of 3, that means 3 meters of leaf per unit area of ground, because that means you're intercepting 90% of the light. If you can do that, you can calculate from these relationships that I've shown you that theoretically you should be able to get up to 160 tons of apple in New Zealand. I did a back of the envelope calculation using these relationships for the UK, and our potential yield in the UK appears to be about 110. That's a long way higher than we are at the moment, and we might in the discussion want to explore why that is. But that gives us, and I think we need to be doing much more of this sort of work, much more of the physics, much more of the mathematics, to complement the exceedingly good biology that we do. And we need to couple that with research on growing systems. Is it actually conceivable that we could intercept 90% of the light? Do we need, or can we, at the moment, we've got quite wide alleyways. We might need those for some purposes, but how far can we go in intercepting more of the radiant energy? And that takes us to growing systems. Pretty well, all of the work that I showed you just now was done with individual trees. Well, we don't grow individual trees anymore. We grow walls and we grow hedgerows. And here in Kent, this is an example of a young orchard that we went and saw last spring. This is apples on a local farm being grown as a wall, staked and wired, branches here, tall leaders, to intercept that radiation. So there's a real challenge there, I think, for us to try and define better what it is we're aiming for. Because if we don't know that, it makes it very difficult for us to have the targets, the right targets in mind. I'm now going to move on to climate and weather. which I think is the next big challenge in this area. Why is that important? Why is it important? Well, I'll try and show you. So, we know from the measurements that have been made here that we are, on average, around 1.5 degrees warmer here now, on average. The air is 1.5 degrees warmer now than it was in the 1960s. Those are the measurements. And that's actually pretty common across most of England. So we know that it's got warmer. The rate of warming, as you can probably see, has slowed, and there are now explanations for this, has slowed over the last decade, but it's irrefutably warmer now than it was in the 1960s. Why is that important? Well, it's important because it means that many, not all, but many of our fruits, and this is Egremont Russell, grown on an M9 rootstock, many of our fruits are now, as you can see, this is the date of flowering, full bloom, which back in the early 1970s was around the 11th of May, and now it's around the 22nd of April. So it's at least 2 weeks, in this variety, it's at least 2 weeks earlier than it was. Why is that important? Well, it might be advantageous, but actually the date of the last frost hasn't changed. So many of our flowers, many of our varieties are going to be susceptible to frost damage because of the earlier flowering. It's important too because the winters are generally warmer, and as those of you that grow perennials will know, the winter cooling, basically the plant uses the cooling in winter to reset its physiological clock. It basically resets a whole lot of biological and biochemical processes, and if that doesn't happen, you get a very funny looking plant. This is a blackcurrant when I was up in Scotland. This is a blackcurrant in the very warm winter of 2009, and you can see that what you get is some buds have broken, whereas other buds are only just beginning. And you get these very odd looking branches on a blackcurrant bush. It also happens in other fruits too. So that warming can disrupt this, or the lack of chilling can disrupt the physiological processes in fruit trees. It's relatively easy to get over by choosing varieties that don't have as much chilling requirement, but it takes time. It takes 12, 15 years to find new commercial varieties with reduced chilling requirement. Thank you. The warming has also encouraged the arrival of pests. This is spotted-winged Drosophila. It's not just down to warming, but it certainly means it's easier for the thing to survive. And that is a big issue now for the industry. But the other thing which is going on, and the other thing which we need to get our heads around, is that very often we talk about the averages, and I've just talked about the averages. But what is also happening, and this is a sort of computer model to illustrate this, but in many people's minds, we know that temperatures, and in fact this is winter rainfall. Winter rainfall varies from year to year. This is the mean rainfall. What's going to happen in the future? Well, if you read the newspapers, wetter winters. So the mean changes, and people assume that this variation moves with the mean. What's obvious now from the various models that the Hadley Centre are doing is that it doesn't work like that. When you run the models, not only does the mean change, but this variation, the shape of this variation also changes. So you'll see here that in some point in the future, I think this is 2080, the mean rainfall in the winter increases, so it does get, the winters are likely to get wetter, but the variation has changed, so we'll get some which are incredibly wet, but if this is the line that marks drought, we get quite a lot that are droughts. Now that's important if you're a perennial crop. If you're going to be there for 25 years, you're going to experience a much wider range of weather. And so we end up in the paradoxical position where that in the future, the winters on average will be wetter, but there'll be some that are much drier than we've experienced. So we're moving into regions, areas of uncertainty that we've not had to deal with before. We've seen some of this winter, quite a lot of this. You probably drove through our version of it to get here today. A lot of waterlogging. Is this connected with the changing climate? Very difficult to ally a particular weather event with the changing climate, but the people who really know about this thing are the insurance industry, because it means a lot of money to them. And in particular, not just the insurance industry, but the reinsurance industry, and the biggest reinsurance industry is in Munich. It's called Munich Re. And Munich Re have taken a particular interest in looking at disasters, natural catastrophes across the world. And you'll see that from 1980 through to 2010, which is covered by this slide, the geophysical events, the earthquakes, the tsunamis, the volcanic eruptions are constant. The thing that has changed is the number of meteorological events, that's storms, and the number of hydrological events, that's floods. This is now increasing considerably, and the economic cost of that too is rising very fast. This is basically showing the same thing, but basically we're on a linear trend upwards in the number of weather-related catastrophes. And that, I think, is significant for the way in which we think about the crops that we are going to be producing over the next 20 or 25 years. We can expect our preconceptions. If I were Dr. Dunstan, I would have had a pretty good idea of when winter happened, when spring happened. I would have some expectation that weather varies, but we're going to see much more of that for the future. And that's shown here. This is a particular study that was done. It basically goes back to 1955. It shows basically those areas in the summer that were very hot or very cold, much hotter or much colder than normal. In 1955, and the much hotter bits are shown by the red and the brown. So the much hotter bits only occupy a very small proportion of the globe in 1955, almost nothing in 1965, in 1975 hotter. But now you come through to where we are now, much more red, much more brown. What does that mean? It means that when I was born in the early 1950s, something which was a 1 in 750 year event is now a 1 in 11 year event. That's a phenomenal change in a very short space of time. Okay, what are we going to do about it? Well, pray. No. What are we going to do? We're going to see, I think, in horticulture, the continuation of a trend which has already started. We're going to see growers wanting to have much more control over the environment in which they grow the crop. They're going to do that in a variety of ways. They could do it simple exclusion of, for example, hail. This slide's from a particularly hail-prone area in northern Germany. They're going to try, almost certainly, and control the environment much more than they've done in the past. We've already seen in this country soft fruit production going under plastic tunnels, different systems of production. I think we're going to find much more exploration of that. We're already starting to see cherries and other materials also going into plastic. And we're going to see control of the rooting environment, and we're likely to see, I think, much more going into glass. It's inevitable, it seems to me, that if we get the genotypes right, we're going to want to control the environment, and we're going to want to do that for a whole variety of economic reasons, not least to increase production, but also to reduce waste. Moving on, my final point really, nutrition, health and novel fruits. There is a concern around as to what we've done with, in fact, all of our agricultural products, not just fruits, all of our agricultural products by increasing production. This is a pretty old slide now, but it makes the point quite well. If you increase, for example, phosphorus fertilisation to push up production of raspberries, the phosphorus concentration goes up when you add the fertiliser, but the concentration of many other nutrients within the raspberry goes down. Now that's not important if you're in a society that is eating well and is not susceptible to mineral nutrition deficiencies, but even in this country now, there's great concern about calcium because people have stopped drinking milk to the extent that they did 30, 40 years ago, and much of our population is now short of calcium, and osteoporosis is coming along, particularly for women. But the basic point here is that by pushing up production, in many cases we've reduced the number of desirable micronutrients within our fruits. This is a survey from the United States. I've just put up 4 fruits here. This is comparing calcium in the 1950 and 1999. By and large, it's gone down quite a lot. Phosphorus, not so much. Iron has approximately halved over that period. And given that 30% of the world's population is short of iron, these are significant issues. Riboflavin's gone down, but actually there are other components, other vitamins, where there's been no change at all. This seems to me to be a very important topic that we ought to be taking a look at. We know that many fruits contain flavonoids. There are 6 flavonoid classes, and depending upon who's done an experiment this week, you will read in the newspaper that either one of these is the superfruit of the future or the food of the future, and you can take your choice really as to which one you go along with. I don't think that many people will be just chewing on onions, but you never know. The flavonoids are important as a class, and the different subclasses. We know very little actually, or I've seen very few studies of how these vary from year to year, how they're infected by environment, and how they vary from variety to variety. This is actually something put out from the New Zealanders, mainly because it shows their varieties in a very good light relative to Cox's. But it's important, I think, to know where we are on this graph, to know what flavonoids that we have. And just to end up with, what about fruit generally? What's going to happen? Well, this is how we market fruit in the 2000s, or this is a slide anyway of a supermarket shelf stacked full of apples and pears and so on. Is that what the future looked like? Does it look like apples and pears? And I'm going to suggest to you that it doesn't. That's not where the future lies. The future looks much more like this. Because with this rapidly urbanizing population, what are the most popular fruits currently? The banana, number 1. Why? Because it's ready to eat. It's got a very good wrapper, and you can chuck it away. Number 2, grapes. Because they're ready to eat, they've got no stones in them, and you can eat them easily. So, I love pears. I had a pear for breakfast. But it doesn't half make a mess of your iPhone if you don't wash your hands first. So, we're the, you know, with it, urban male. We know that in our kitchens, when I was first married, on average, women spent 45 minutes of the day preparing the main family meal. They now, on average, spend 12 minutes. And that is not something which is going to change in a hurry. So we want the convenience. So we want an easy to throw away wrapper. In this case, the plastic container provides it. But maybe we could think carefully about what we're growing our fruits in. What about the peel, the skin, and so on of our fruits? Can we modify that to assist with the style of life that we now have. We want it ready to eat. We want it sweet and juicy. The sugar content of grapes has gone up by 25% in the last 20 years. We don't want any pips or stones. So the future for cherries is the stoneless cherry. And we want it to, of course, we want it to look attractive. And I'd like to thank lots and lots of people who've provided inspiration for this. On the left-hand side, I hope I've mentioned everyone whose work I've purloined. On the left-hand side, here at East Malling. On the right-hand side, from other institutions. And of course, I must thank the East Malling Trust for the financial support that they provide to this institution. Not just the financial support, but the moral support too, to this institution. So to conclude, we've actually moved a long way from the concern about individual trees. Rootstocks, to a large extent, played a big part in that. We're now growing hedges and walls. We don't know the basic physics of those. We haven't been able to invest in that in the past. Genetics, environment and management is at the heart of what we do. It will continue to be so, but we'll be adding to it much more multidisciplinary working teams. And finally, it's incredibly exciting, isn't it? What we are attempting to do. This is a great time to be involved with this topic and with this industry. And on a personal note, I'd like to thank the many scientists who work with us and have joined us in the last few years and dedicated their careers to actually assisting with the challenges that we face. And I'd like to thank too all the many support staff that we have here in finance, on the farms, everywhere, who are supporting us in this endeavour. It really is an exciting time to be here. Thank you very much.